So now let's see how the process of transcription continues. First we have seen RNAP comes, or the RNA polymerase comes here. Along with RNA polymerase, there are many transcription factors attached here. So this is our transcribing machinery. Here is our transcription start site. Suppose it's here. So what happens? This DNA molecule actually unwinds in this portion. Due to that, there is a creation of a bubble-like structure here. This portion, this is around 12 to 14 base pairs long and is known as transcription bubbles. In the transcription bubble, rRNA polymerase is transcribing the RNA molecule. So the raw material for transcribing RNA or for the formation of RNA molecule are either adenosine triphosphate or guanosine triphosphate or cytidine triphosphate or uridine triphosphate. Now we have to remember, here are the three phosphate groups present. Then there is our sugar moiety and the nitrogenous base. All the other molecules are present as nucleotides, that is one phosphate, one sugar, one base. Only the first raw material here, it retains all its three phosphates because it's not attached to any other nucleotide before it. So in the beginning we always have three phosphate groups, then sugar and nitrogenous base. Then all the other as nucleotides, phosphate, sugar base, phosphate, sugar base, phosphate, sugar base, because two phosphate groups are removed as pyrophosphate, like this. But from the first molecule, this pyrophosphate group is not removed as it's not attached to any other nucleotide before it. So in the beginning we always have three phosphate groups. Then more and more nucleotides gets attached and the chain keeps on getting longer. When this chain is around 9 to 10 base pair long, then conformational change comes in this RNA polymerase enzyme. Due to that conformational change, sigma factor gets removed and RNAP moves away from this promoter region towards the structural gene, this side. So this process is known as promoter escape, and this is needed for beginning of the transcription or for speeding up of the transcription. After that, more and more nucleotides are getting attached with this molecule and we keep on elongating the RNA molecule. For this process to keep on occurring or for this process to continue, we need the elongation factors. See, these elongation factors can be positive elongation factors or the negative elongation factors. Examples for the negative elongation factors are NELF or negative elongation factor. Second one is DSIF or DRB, sensitivity inducing factor. These two are the elongation factors and they cause the promoter proximal pausing. What is the promoter proximal pausing? This RNA polymerase, after the promoter escape, tends to halt again and again. So that is called promoter proximal pausing and is caused due to the negative elongation factors. To escape the promoter proximal pausing, we need the positive elongation factors. Example is PTEFB or positive transcription elongation factor B. This factor causes phosphorylation of these two negative elongation factors and nullify their effect, and also it causes the phosphorylation of various amino acids in C-terminal domain of the RNA polymerase II. So, that increases the processivity of the RNA polymerase. So, this is the main positive transcription elongation factor B. What are the other examples? Other is TF2S or transcription factor 2S. This is elongation factor and it helps in the RNA polymerase to bypass the transcription blocks. What are transcription block? See, after covering this region which was proximal to the promoter region, this region, all along the path, this RNA polymerase tends to slow down or tends to halt again and again. These are known as the transcription blocks and these are bypassed with the help of transcription factor 2S, which is actually a elongation factor. And what are the other elongation factor? One more example is the FACT, facilitates chromatin transcription. This FACT is actually a protein complex that partially disassemble the nucleosomes ahead of the RNA polymerase and reassemble them after the transcription passes. So, when the nucleosomes are disassembled, that makes the DNA molecule available for the process of transcription. So, this is one elongation factor. It facilitates the elongation. This transcription factor 2S actually bypasses the transcription blocks along the way of elongation, and this positive transcription elongation factor B actually helps to nullify the promoter proximal pausing. So, these are the negative and positive elongation factors. And more nucleotides keep on coming and they keep on getting attached with this chain, which was made around 9 to 10 base pairs long before the promoter escape. So, because this transcription start site actually occurs in the promoter region only. So, once this chain is 9 to 10 base pairs long, then promoter escape occurs, then there are chances of promoter proximal pausing and various transcription blocks along the way. These are overcome with the help of many positive elongation factors.